why don't we take a second and take a couple of deep breaths and stretch a little bit and move in our seat in here and get ready for continuing that conversation about all of the connections that our plants and soils and insects and the world has and shares, and get ready for Linda to do her talk about roots and the environment below ground. Linda, thank you.
Well, good afternoon, morning, whatever. Sorry. Good morning again. As I said, it's great to be here. I really enjoy this group, and I've had some wonderful conversations with some of you after talks and during dinner, and so just love being here. There is a handout for this, so if you haven't got the handout, make sure you wave your hand so it can be delivered to you. It's got the notes from the talk as well as links to all the websites that you've already heard about yesterday, but I just want to make sure you've got that. Okay, there we go. So, as Brian said, this is the landscape below ground. So, we talked a little bit about roots and mycorrhizae and nutrients, a lot about nutrients yesterday. And this is looking at everything that's happening underground. So, back when I started doing my horticulture studies, and I'm not going to tell you how many decades ago that was, there wasn't really that much we knew about what was going on underground because it's so hard to study roots in their native environment. As soon as you dig them up, they're no longer in their native environment. So with the invention of things like rhizotrons, where you have these underground windows and you can use pretty non-invasive techniques to study roots and movement and microbes and everything else, we're learning a lot more. So this is really interesting stuff, and most of it, if not all of it, is things that I never learned about when I was getting my PhD. So one of the things we have to think about as we're starting to go underground and see what we can see is understanding root structure and function. So root structure normally looks pretty much like this picture right here. Roots are like spokes on a wheel, the structural roots are. And the fine roots extend from those. And when they're in the landscape, the same thing. You're going to see those spokes on a wheel. They're the structural roots. They hold things in place. They are not absorptive roots. They're structural roots. They're storage roots. They're for stability, but they are not for uptake. So the only uptake is really with the fine roots, and so we're going to be focusing on those. But what we need to think about when we're talking about tree roots is that a woody plant is a woody plant. And except for really unusually adapted plants, like a lot of xerophytes, as a matter of fact, they're pretty much the same. A tree is a tree is a tree, and their physiology is remarkably similar as you move from species to species. There's not that much difference in the basic physiology. As I said, with some things, especially like wetland adapted plants or things that can adapt to toxic soils, there's a little bit of difference there, but in terms of structure and function, it's pretty much the same. The fine roots are the absorptive roots, and we don't see those because they're really hard to see. And the only part that actually absorbs is where the root hairs are, which is at the tips of the fine roots. And so that absorptive surface is really a very small part of roots. And we see the things that are fine roots, but not necessarily the root hairs. You can see them if you make cuttings. This is the easiest way to do it, is to make a cutting. You put the thing in the water, you're going to have all kinds of roots come out, and you can finally see those root hairs. So the fine roots do uptake. They do growth regulation. There's all kinds of growth regulators that are produced in these actively growing root tips. They will increase in length. And as they increase in length, the absorptive capacity stops at that one point because the growth is past that point. So they become they still are transporting things, but they're not actually taking things up from the soil. They're transitory. They're really very opportunistic. And what I mean by that is that if there are resources, if there's water and nutrients and oxygen, they will grow into those pockets. If the water and oxygen disappears, those roots will die, and then other roots will grow. So if you're watching these things in a rhizotron, you'll just see all this movement where things are going ahead, and they stop, and other ones move, and so they're kind of back and forth, always following the nutrients, water, and oxygen. So as I said, they're opportunistic, they grow where they can, and they interact with microbes. So when we're talking about the mycorrhizae and the beneficial bacteria, they are interacting with these actively growing transitory roots. Okay, storage and structural roots have a completely different function, as you might expect. They don't take things up. It's not that they can't develop new roots, because often they can, especially if they're pruned or damaged. New roots can arise from beneath those damage points. So they can send out new roots, but as a lignified woody structure, they're not uptake roots. They do increase in girth, much to many of our clients' unhappiness as they emerge from the landscape and start getting in the way of mowers and things. And people always wonder, why is all of a sudden all these roots coming up, not realizing that they're not only increasing in length, but they're getting wider and wider and wider. And they'll be pushing up as well as pushing down. As I said, they're woody, which means they're lignified. Once they are lignified, they do not change their form. It's like trying to bend a piece of wood. So this gets a lot into when you're transplanting and you're looking at root structure. If you have woody roots that are circling, they're not going to all of a sudden straighten out. So a tree that's in a bag or a container that has a circling woody root, it is always going to be circling. It is not going to grow straight. So it's one of those things will either become a non-functional girdling root, or it has to be pruned away. And these roots are really resistant to stress. No big surprise, right? Because they're lignified, they're superized, they don't lose water. It's hard for disease to get in, so they're very resistant, which is good because they are, basically the stability organs of the tree. So as we're looking at roots and as they age, this is a really important point here. And many people still don't realize this, that there are juvenile roots and there are mature roots. A juvenile root has vertical growth because its function is to grow down as straight as it can to stabilize the developing tree and to keep it in place, right? And that's the taproot. So all juvenile trees, because there aren't any real, well, if you want to consider a palm tree to be a tree, you can. It's a monocot. But all dicots have this taproot, right? So you learn this in basic botany. But then unfortunately, we kind of think that a mature tree has this giant taproot. It does not. A mature tree does not have a giant carrot at the end of it, because the root's function after stability is expansion and getting nutrients and water and competing. And it's not downwards where growth is going to go, but outwards. So this taproot on mature trees does not exist. And we have to remember that because there's these perceptions that certain trees have these big taproots and you can't transplant them. Absolutely not true. Oak trees are often given as an example of trees with taproot that you can't transplant. But did you know that when oak trees are commercially propagated, they're pulled up, the taproot is cut to encourage lateral branching. So the taproot is a juvenile structure. It's not critical to a mature tree's health. In fact, it doesn't exist in a mature tree. So a mature tree has horizontal root growth. I know you've all seen this picture before. It's famous among arborists. It was as a diagram from many, many years ago. But it shows you, most of the growth is going to be horizontal. Oxygen is close to the surface. Water is coming from the surface. Nutrients are at the surface. That's where the root growth is because that's where the resources are. Occasionally, you'll find little sinker roots, as you can see in that picture where you have roots growing down. Those are not taproots. The taproot is a juvenile structure. But you do get sinker roots. And so the sinker roots occur wherever a growing root finds a pocket of oxygen and water and nutrients, and it will grow into that. And so they are vertical roots, but they are not taproots. So it's really important we explain to our clients that the root they see on a young tree, a taproot, is not something that persists into the mature tree. Okay, so now we want to switch to soils and nutrients and things like that. And last year, I know you had James Cassidy here. Those of you that were here, isn't he great? He's a wonderful speaker. I just love him. But anyway, I'm going to just review a little bit about soils. You have sandy soils around here, mostly, right? Except where you're near water, and you might have other types of inorganic material there. But sandy soils don't hold much in the way of nutrients. They don't hold much in the way of water. They have lots of oxygen because their nice, coarse texture allows oxygen to move through easily, but also means water moves through. The reason they don't have much in the way of nutrients is because sand is not a charged particle. Okay? It's neutral. So things don't bind to sand. So nutrients can just wash through unless there's organic material in the soil, either within the soil or as a mulch on top of the soil. Silt's kind of the same way. It's a smaller particle size, but it's not charged, so it doesn't bind nutrients. It doesn't hold water, but it doesn't have much oxygen because it's kind of like flour. If you were to rub silt in your fingers, it has a very floury texture. So it's still a round particle, but it doesn't really have much in the way of resource utility. The utility comes from the organic matter that hopefully is part of the soil structure. The clay, even though we say we hate clay, we have to have clay. And the reason is because clay is negatively charged and it will bind to cations. So your cation exchange capacity in a soil is very much dependent on clay and organic matter. And you have to have a clay component to have a soil that will bind clay, the soil that will bind nutrients without necessarily having much organic material. So the clay is really crucial. So, you know, a good loam has parts of all of these nutrients in it. So the clay has high nutrients, but very little oxygen because rather than being round particles, clay is flat particles. So as you can see from that picture on the right, very flat particles. So very little oxygen, not very good water movement. A clay soil is really tough for plants to deal with, especially if it's compacted. So what happens in looking at resource availability and the way that soils are structured? So unfortunately for us, when we're looking at developed areas, in urban areas or residential areas or whatever, we have layered soils. And this is because when development happens, topsoil, whatever topsoil exists, is scraped off, and you're going down to the subsoil, and it's compacted, and you build on it, and now you have all these layers of stuff. And so it's not a really functional soil because it's hard for both water and oxygen to move through those layers. So the more layers you have, the harder it is on water and oxygen movement, and the harder it is for roots to grow. Compacted soil is kind of the same type of problem in terms of poor water and oxygen movement because now the pore space has been squished. So even if you have, you know, a relatively sandy soil and it gets compacted, it's going to have less and less pore space, which means, again, roots have a tough time because they can't follow water and oxygen because it doesn't move through this. So those are two things that will make resources very unavailable to plant roots because of the lack of pore space or because of those disruptions from the layers. Mulch soils have the best in terms of water movement and air movement because the mulch protects the soil from being compacted. And even a compacted soil on its own can loosen just by having a mulch there because the living organisms that are still in there will loosen the soil once it's protected from further compaction. So a mulch soil will recover on its own as water can move in, and when water moves in and gets through those layers and through those particles, you start getting oxygen, you start getting life again. So that's what brings life back to a compacted soil. Okay, so what do roots need? They need water, always water. They need nutrients. We all know these ones. Sometimes we don't remember that they need oxygen. And a lot of your clients might think because plants produce oxygen by photosynthesis that somehow they get the oxygen to the roots. Unless it's a wetland plant, oxygen is not transported to the roots. The roots have to get oxygen from the soil. So if the soil is very low in oxygen, then the roots aren't able to do their various metabolic activities, right? Because they don't have any oxygen. And then the last group are the microbial partners. And they're not necessarily essential, but they're necessary for trees and other plants to have optimal growth. So those microbial partners really are important, especially if you're in a harsh environment, to keep your trees healthy. So this is going to kind of go back to a previous slide looking at how water and oxygen move, but we have to kind of shift thinking now and think about what impairs soil and root function. So as I mentioned, layered soils, you're going to have poor water and oxygen movement, so that's just something I showed you just before. Amended soils have the same problem because an amended soil acts like a layered soil. So if you work materials into the soil when you're planting or when you're building or whatever, you're creating layers. And when you have these layers, whether they're vertical or whether they're horizontal, water and oxygen don't move through them. And so you'll have water puddling on top of landscapes that have amended soils because they hit that barrier between the amended and unamended soil and the water stops moving. Compacted soils, as I mentioned before, low oxygen and water, and poorly mulched soils, those that are mulched with sheet mulches, also have reduced water and oxygen movement, as I mentioned yesterday when we were talking about various types of mulches. So a lot of these things are happening in our urban environments, and we have to realize that means that oxygen and water levels may be very poor. Even in a sandy soil, you may have very poor oxygen and water movement. Okay, so now we're going to get into the microbes and talk about them in general and then with some specifics. So generally, the beneficial microbes, these are relationships that evolved a long time ago. And the older the plants are, in terms of evolutionary time, the older the relationships are that they have developed with microbes. So they're mutualistic in that each party benefits. So the little cartoon there is showing arbuscular mycorrhizae that is giving the plant mineral nutrients, and the plant is giving the fungi sugar. So that's kind of just a little cartoon to show how the relationship generally works. So as I said, the plants are receiving water and nutrients, and even though plants can take up water themselves, mycorrhizae are much more effective because their hyphae are much thinner and longer than root hairs are. So it's like root hairs on steroids as being this big mass of very fine hyphae that are able to take up water and nutrients. The microbes get sugars. They also get B vitamins. They probably get other things too. The more we study these, the more we find out. But this is just a really general talk, so I'm not going to go into all of the little intricacies. And the cool thing is that these are found throughout the plant kingdom. And 60 or 70 years ago, when we were first discovering the presence of mycorrhizae and realizing that not all fungi are pathogens, this was thought to be kind of an anomaly, that we've got some fungi here that are beneficial, and the more they were studied, the more it became the rule rather than the exception. So many plants have mycorrhizae. Nearly all woody plants, if not all woody plants, have mycorrhizae. Some of them may be facultative in that they require the mycorrhizae. Some of them may be facultative means they don't require the mycorrhizae, but some are obligate. They have to have the mycorrhizae to survive. So this is why it's kind of a confusing area of study, because sometimes when you study a plant, it may not have the mycorrhizae because it's facultative. It doesn't have to. The obligate ones we know about. They're easy to study. Okay, so what are mycorrhizae? I love to break words down because it helps remember. Myco just means fungus, and rhizae means roots, so this is literally root fungus, or fungal roots, and it says exactly what they do. They're usually mutualistic. I'll mention when they're not. So in other words, they're both benefiting from the relationship. Sometimes they don't. They're in all woody plant families, so all the families of woody plants have members that have mycorrhizae. They really need a source of decomposing wood to function optimally. So this is why I talk so much about the importance of woody mulches, because the woody mulch provides that source for the mycorrhizae, because mycorrhizal fungi are also decomposers. So they get a lot of their nutrients not from the soil necessarily, but from infiltrating, decomposing wood and extracting nutrients for them. Okay, so first of all, we've got the endomycorrhizal fungi, and sometimes called arbuscular mycorrhizal fungi. It's a big mouthful. AMF is an easy way to talk about them. So these guys are found inside the plant roots. You don't really see their presence so much on the outside, because most of what they do is on the inside. So they actually infiltrate all the way into plant cells, not just kind of around and into the dermis of the root. And this is why they're called arbuscular, because when they get inside, they create this structure that looks like little tiny trees. So arbuscule means small tree. And so you can look at micrography of this, and they're so pretty. They look like these cute little trees. And that's because they get in there and they put their hyphae out, and that's their absorptive surface where they're taking things from the plant and giving things back to the plant. These guys are obligate symbionts. So as I said, some plants require mycorrhizae and some plants grow with them or without. Some mycorrhizae have to have plants or they don't exist. And this is true with a lot of really interesting fungi. There's some that are probably in the process of becoming part of the plant. So not to get too far afield, but you probably know that chlorophyll have chloroplasts in them. The chloroplasts are remnants of free-living algae that eventually became part of the plant genome. And the reason we know this is because if you look at the DNA in the chloroplast, it's different than the rest of the plant's DNA. Same thing with mitochondria. You probably have heard of mitochondrial genetic analysis to find out your mother's. It follows the maternal line. This is in everything that has mitochondria. The mitochondria are also remnants of free-living bacteria. So it's really cool that this has gone on and on, through eons, and that certain microbes become part of a larger living organism. It's really fascinating. So anyway, since they live within the plant, they're hard to study. They have never seen sexual reproduction occurring. So they just reproduce vegetatively. They may reproduce sexually, we just have never seen it before. The earliest known species are in the glomerulomycota, and those are the first ones to have been apparently colonizing the earliest plants. They're very common in mid to low latitudes, so we're getting down here in New Mexico where you would find them. They don't have as many of them up where I live in the more temperate areas, and they are very widespread through hundreds of plant families because they are the first ones. And so they colonized early on. They got spread through different plant families as those populations were evolving themselves and creating new species, and so they're very widespread. But they are things that occur mostly in the tropical to the subtropical areas. Now, so those are the ones that you can't see easily. The ones you can see easily are the ectomycorrhizal fungi, or EMFs. And ecto just means outside. So when you see these hyphae, these kind of cottony sheaths around roots, you know you're seeing an ectomycorrhizal fungi. They live as decomposers by themselves or symbiotically with tree roots. So it just depends on the species, on whether they're going to require the host or whether they can also just be free-living decomposers. So you'll find them just oftentimes in mulch piles. And you'll see many of them come up with their interesting little fruiting bodies. Sometimes the famous one is the dog vomit fungus, which always seems to grow on top of mulch piles. But you can see their presence in mulch piles. They will reproduce sexually and asexually, so the sexual part is the part that comes above ground and that we know as mushrooms or toadstools or whatever. And not all mushrooms and toadstools are mycorrhizal, but all mycorrhizal fungi will have some kind of above ground fruiting body. So these are ascomycetes and basidiomycetes. So if you're a mycologist, which I am not, those will be familiar terms for you. And they're at all latitudes, but only in 3% of the species. And that's because it's a newer relationship in terms of evolving to become part of the plant's physiology, really. And so there's just not as widespread. Over time, you'd expect to find them in more and more plant species. And by over time, I don't mean in our lifetimes, but just over centuries, you might find these expanding. So in general, this is how roots are colonized by mycorrhizae. So you've got spores everywhere. We know this, right? You have beneficial fungal spores, you have pathogenic spores. There's spores everywhere in the water, in the soil. Within the soil, they're everywhere. They may not be active, but they're present. So the spore repositories for mycorrhizae are generally most prevalent in really coarse organic material because they are decomposers. They germinate when it's moist and aerated, but they also have to have a receptive root that allows them to become inoculants, okay? So if they're going to become mycorrhizal, they have to have that cue. If they're just going to be free-living decomposers, they just require moisture and aeration. If they're going to be mycorrhizal, then the root has to send out a chemical signal to invite them to inoculate them. So the hyphae grows by the roots when this happens, and then they either become, depending on what they are, they either become just basically internal mycorrhizae, or they're the ectomycorrhizae. Many of these have multiple hosts, but some of them are very specific. Some plants are very specific for the mycorrhizae, and some are very general. So it's very complex and hard to predict exactly what's going to happen unless you study them very, very carefully. With AMFs, the colonization, the picture that kind of shows what happens. So at the top there, you can see those yellow threads, and that's where the hyphae are entering the cell. They go through, or the root, they go through the epidermis, into the hypodermis, then into the cortex, and they end up in various areas where they may form vesicles, which are full of lipids. And the lipids are something that they use and can take back into them because the plants will make the lipids. Some of them are they're taking sugars away. The ones that look like little trees are absorbing sugars and things like vitamin B. And it's also where they're putting the water and nutrients. So those are the sites of exchange. So they're inside the plant. They're becoming, kind of plant organelles, right? So that's where nutrient exchange occurs, as I mentioned, and then those vesicles are for the lipid storage. So it can be very complicated. It's like an apartment building with all these various things going on within the roots. And this is something that happens with the majority of angiosperm species, about half of the gymnosperm species, and with about half of all ferns and other kind of more primitive plants. So they're very widespread. There's a group of plant growth regulators which did not exist in our knowledge when I was in college. So I'm telling you really how old I really am. And there's a new group called strigolactones. So in addition to auxins and cytokines and things like that, there's strigolactones. And strigolactones have a special function in inviting the mycorrhizae to colonize them. So the strigolactones are released from the roots. This allows the hyphae to penetrate the roots. And during this time, a really interesting thing happens to the roots. The roots stop growing laterally in terms of having new little lateral roots come out. They look stubby, and you can't see root hairs on them. They just look like fingers or like coral or something like that. They don't look like a root system. They look strange. And a lot of people used to think they were diseased or something was wrong with them, but they're not. It's because now the plant's not putting resources into nutrient uptake because it has the mycorrhizae to do it. So they save the energy of growing root tips, and a lot of fine roots because they've now got this fungal partner that does that work for them. They have to have low soil phosphate to do this, okay? So if there's high soil phosphate, we talked about this yesterday, the plants do not release the strigolactone, and you don't get the inoculation of the roots. So there's this inverse relationship between the strigolactone release and phosphate levels. The higher the phosphate level goes, the lower the strigolactone release goes. Okay? So this is why it's so important not to have overly rich, well, not to have excessive amounts of phosphate in the soil, because you will reduce this colonization, which is overall so beneficial for root health. Okay, so with the EMFs, it's a little different because the hyphae grow in between the cells. This relationship, I would hypothesize, if you give it enough time, eventually they will probably penetrate and grow into the cells, just like the AMFs have. But right now, this is a newer evolutionary mutualistic relationship. And so right now, they grow between the cell walls, and they create a sheath, kind of a cottony sheath. And some of these are going to the cortical tissue, so now I'm testing you on your understanding of root morphology. And some of them are epidermal. Again, this is kind of following an ecological or an evolutionary trend. The cortical tissues are deeper in there. This is a relationship in gymnosperms, an older group of plants. It's been going on longer than the angiosperms, right? So you can kind of see how very slowly things go from being kind of an external partner to becoming an internal partner given enough time. And like with the AMFs, the root hair growth is inhibited during this time because the hyphae are now going to take over that role. So you don't see root hairs so much, but you see lots and lots of very fine cottony material, which are the hyphae. So EMFs tend to be fairly general in the things that they will inoculate. They're not really that specific. There are many stimulants besides the strigolactones that are released by the plant roots that will allow them to germinate. But just like with AMFs, they have to have that low soil phosphate. So no phosphate fertilizer unless you have absolutely no phosphate in the soil. But you can see how odd, especially the roots on the left look. They look kind of contorted and strange and not very root-like. And it's just because they're not putting all their resources into uptake roots anymore, because they don't have to. And you may have heard the wood wide web, which is a great tongue twister, before. And this is kind of the hypothesis that everything is connected underground through roots and mycorrhizae and all these relationships that we didn't know about, 100 years ago or 50 years ago. So the hyphae extend throughout the mulch and the topsoil, okay? One of the reasons why wood chip mulches become locked in to the soil system because the hyphae are holding them there. So they're going throughout the mulch and the topsoil, and then they're connecting to the roots underneath. And so they're anchoring the wood chip mulch and feeding from it as it decomposes. So they actually decompose that material. You can easily see this if you get into a mulched area, move some of the drier mulch aside, and you will see all those hyphae in there, and a lot of them wrapped around roots. Excuse me. So they take up nitrogen, they take up phosphate, probably take up other things and water much more effectively and efficiently than roots do. And this is why it's a good partnership. And then you see the fruiting bodies, and that just shows you where your actively growing roots are. Because remember, these grow in actively growing roots. So when you see these fairy rings, which I'm old enough to remember when everybody said those were disease organisms, when the mushrooms making the fairy rings, those are just the areas where the fine roots are, because that's where the fruiting bodies are. So you can watch your root system growing underground if they happen to be mycorrhizal, because you'll see the mushrooms moving out and away from the tree itself. So what do mycorrhizae do? Well, they penetrate pores in the soil that are much smaller than what root hairs can get into because they're much finer. So they can get into spaces where roots just can't grow. They solubilize rock. They release acids from their structures that will make it more acidic, so they're solubilizing the rock using protons or hydrogen ions if they're AMF, and small organic acids if they're EMFs. So they're helping kind of literally melt the rock, make that inorganic material available for uptake, and this is how pioneering species can colonize on rocks, pioneering plant species, with the help of these microbes. And they both respire. Okay, they, just like we do, they take in oxygen, they release carbon dioxide, and carbon dioxide can become carbonic acid, right? And that will also acidify the environment. So those are the sources of acids that help break down the rock. So mycorrhizae will increase root interconnectivity. They mineralize the soil, and they will bind up and sometimes kind of detoxify heavy metals by turning them to a form that's not as toxic, and that protects the plant roots. They will stabilize soil structure by increasing the aggregation of soil particles, because again they're kind of growing into and connecting soil bits and making PEDs, and we've all seen those structure, the chunky structure which says you've got a really healthy soil. And they also enhance the formation of slowly decomposing organic matter, or what I like to call slow food when it comes to organic matter, as opposed to fast food, which is like compost. Okay, so slow organic matter just decomposes at a much slower rate. Sometimes it's called humus, but humus implies that it never gets degraded, that it stays in that form, and that doesn't happen in nature. It happens in the lab when you treat it with a reagent at 12 pH, but it doesn't happen in nature. So it's another one of those things that we can go off into a rabbit hole talking about myths, and humus does not exist in nature. This is where the beneficial relationship sometimes falls apart. So mycorrhizae don't do their services for free, right? They're taking sugars, they're taking vitamin B and perhaps other types of nutrients from the plant. And so they will take generally somewhere between 5 and 10% of a plant's energy, sometimes as high as 20. So it's kind of a high tax. But on the other hand, the plants do extremely well, and it becomes something that if they're healthy plants, they can tolerate. Now, this becomes a parasitic relationship when plants are in bad shape. Now, that doesn't mean that you should fumigate your roots and kill off the mycorrhizae. What it means is you've got to get your plants in better shape, whether it's through more water, mulching, whatever, planting correctly, all the reasons that plants don't survive. But this can actually, the mycorrhizae can actually hasten a tree's demise if the tree is not in good shape. So it's just something to keep in mind that even though mycorrhizae are really a tree's best friend, if the tree is in some kind of mortality spiral, mycorrhizae can just speed up that spiral. Okay, so that's a little bit about mycorrhizae. I do want to talk about beneficial bacteria, or sometimes what are called plant growth promoting bacteria, so PGPBs. That sounds, never mind. It sounds like that club in DC that was part of a Talking Heads song years ago, and now I'm really showing my age, but we'll leave that alone. The group that we know the most about are probably the nitrogen-fixing bacteria, right? We're familiar with those, things like rhizobium. And we kind of think of legumes, peas and beans, but a lot of other plants, including trees, are nitrogen fixers. Alders are a good example. And if you go online and even just type in nitrogen-fixing trees, you will find a whole bunch of trees that are nitrogen fixers. Again, we've known so little about the landscape underground for so long that except for crop plants, we really don't know how many trees are nitrogen fixers, but many of them are. There's also free-living beneficial bacteria that are not associated with becoming part of the root morphology, and they fix nitrogen just naturally in the soil. So they will bring nitrogen into the soil, and nitrogen being a limiting nutrient, this is a really positive relationship to have those free-living bacteria there as well. And then there's also some free-living bacteria such as bacillus. So all of these things can fix nitrogen, which when it goes from gas to solid form, becomes available for plant uptake. So the symbiotic nitrogen fixers, you've seen those if you've looked at, again, any kind of beans or peas, and you pull them up and you can see those nodules. You cut into them, and it's kind of weird because it looks like they're bleeding because they have a pigment that's very much like our hemoglobin that attaches to oxygen, okay? So this is called leg hemoglobin, and it attaches to oxygen, which is important because it keeps the oxygen away from the nitrogen fixing process. So the enzymes that fix nitrogen are disabled by the presence of oxygen. So the leg hemoglobin, it attaches to the oxygen so it doesn't kill the enzymatic reaction. It's really interesting. There's free-living bacteria's biofilms. So everyone's familiar with probiotics, right? And the whole probiotic craze that started, I don't know, 10, 20 years ago, about eating yogurts and all these other things are really good for your digestive system. So you're familiar with those and how those will help keep pathogens from becoming a problem in your digestive tract. So this is kind of like this, except it's on the outside of the roots, where they form a film that prevents pathogens from attaching. So the bacteria are, again, they're attracted by organic acids released by the roots and sugars. So there's something to this whole notion of adding sugar to the soil, because this could help with getting beneficial bacteria to make a biofilm. So this, again, Dr. Glenn Percival, who's one of my favorite researchers, has done research on this and looking at adding sugar. And while adding sugar to soil itself probably wouldn't do much, there is evidence that if you have a microbial-rich soil with plant roots in it, that it might increase the biofilm production. Don't know yet. It's a very interesting thing. The other thing that'll happen with these biofilms is they will suppress the plant's immune response, which they kind of have to do, right? Because it can be perceived as an infection by the plant, and it's the same with the mycorrhizae. It's perceived as an infection, and then the plant's defenses will try to fight it. So they suppress that system as mycorrhizae do, so that the plant doesn't reject them. So that's an important part of this, is that these microbes keep the plant from reacting in a negative way to them. So these free-living bacteria, these biofilms, they're going to be attracted by those. Whoops, I think that's the same slide. How did I do that? Whoops. Sorry. Here we go. Okay, so what the PGPBs do is they prevent colonization by the pathogens because they're taking up space. That's an obvious one. And maybe a not so obvious one is that they actively destroy they can actively destroy pathogens by producing cyanide and degrading fungal walls and doing all these other really active types of defense. And so you may be thinking, well, what happens to the mycorrhizae, which are fungal? And that's a question I can't answer for you. I mean, I'm assuming that there's some way that those are protected, but I don't know. And it's because there's so much research on this now that I haven't even been able to keep up with. I'm not sure that question's been answered. So if any of you have read something that explains why mycorrhizae are not degraded by beneficial bacteria, I would love to talk to you. Okay, so beneficial microbes will increase soil organic matter content. They will solubilize phosphate, as I mentioned before. They will increase uptake of nutrients, as we talked about. They will fix nitrogen, many of them will. They will produce stimulatory plant hormones, which is always beneficial. They increase leaf, root, and shoot growth as part of this process, and they will improve plant survival and establishment. There are tons and tons of research papers on this topic. So this is really very exciting in terms of our understanding of how important the microbial community is to healthy root systems. They're seen to improve soil structure. They will improve competition for nutrients and water among plants. So a mycorrhizal plant, for instance, will be more competitive than a non-mycorrhizal plant just because they've got a bigger and wider web of hyphae to take things up than non-mycorrhizal plants would with their roots. And they will increase resistance to many environmental stresses, which makes sense. The more hydrated a plant is, the better its nutrient content, the more it's able to put compounds into defensive compounds to then resist environmental stresses. So what inhibits these guys? And there are things that inhibit. So this is the take home for your clients about what not to do if you have, if you want to have a healthy microbial community. Soil fumigants should never be used anyway unless you happen to be producing strawberries or something else in a monocultural system where your goal is to increase and maximize yield. But that's not our goal as landscape people. So we don't ever want to use soil fumigants. We don't want to use excessive fertilizers. We talked about yesterday, especially phosphate, because that just screws up the whole invitation system that the plant has to allow it to be inoculated. Excessive irrigation. And I don't mean, now excessive is kind of a weird word, but this just means you don't want waterlogged soils. The amount of water you put on a sandy soil may be much more than you do onto a more clay soil, but you don't want standing water because no oxygen. So you don't want excessive irrigation or poorly drained soil, poor soil structure. So when you have these problems with amended or layered or compacted soil, it doesn't do well, again, because of lack of oxygen. And tillage can ruin these PEDs. And unfortunately, lots and lots of people think that a healthy soil looks like a handful of coffee grounds. A healthy soil looks like a big chunky mass of stuff with hairs coming out of it. And that's what a healthy soil looks like. It does not look like coffee grounds. Whoops, keep doing that. Topsoil removal obviously is going to cause a lot of damage to the living part of the soil, and that's why it's so important, if you can, when things are being developed, whether it's a park or whatever, that the topsoil can be retained and then replaced after everything is done, because that's where the life is. And compaction. So those things will all inhibit your beneficial microbe community. Things that stimulate microbes. Big surprise, coarse woody mulches will stimulate both really the fungal community, but at the same time, because it's increasing oxygen and water holding capacity, you get the beneficial bacteria as well. Conservative levels of nitrogen. Remember I said yesterday, if you have to add something, add some nitrogen, because it not only helps the plants, it's going to help the microbes as well. So microbes also, as their limiting factor for growth, nitrogen is it. Warm temperatures, you can't really, unless you can control the weather somehow, you can't really guide that, but you can keep soil mulched, which during the winter will keep it warmer. And you can also keep it mulched in the summer, which will keep it cooler so it doesn't get hot. So mulch has a moderating effect on temperature. It stays warmer in the winter and stays cooler in summer. That's good for the microbes. Mild drought or nutrient deficiency, which seems odd. But again, when you have low phosphorus, that's what sends out the signal to the microbial spores, the mycorrhizal spores anyway, to go ahead and germinate and come and inoculate me, right? Because low nutrients are what the plant is experiencing, so that will help increase the inoculation of the plant roots. Reduced pesticide use will help. Reduce soil disruption. So you see here that we're looking at kind of no-till stuff. You're not trying to, not a lot of things added, trying to keep things as natural as possible. High plant diversity, just because different plants have different requirements for different types of microbes. So the more diverse of a palette you have, the potential for microbial diversity also increases. So living mulch will help if you can. In a desert environment, living mulches are kind of tough to manage because they require water. But for a lot of you that are managing gardens and other public spaces where you do have irrigation, a living mulch will help. And obviously strongly mycorrhizal plants. Woody plants are great for microbial diversity. Just because they've been around a long time, they got massive root structures, and they just have the ability to create these very diverse beneficial populations of fungi and bacteria around them. So we talked about this a little bit about packaged microbes, but I figure we might as well do it one more time, just so you're thinking about it. What about these inoculants? And not just things for mycorrhizae, but you can get probiotic inoculants as well. So there's been a lot of research looking at how effective they are. I mentioned yesterday it's impossible to tell whether the spores are alive or dead unless you have a microbiology lab to plate everything out to find out. Now they've gotten really tricky. So going back in time, 20 years or so, when they first started with the mycorrhizal inoculants, they were just the spores. Now, if you look at the packages, they contain nitrogen. So you will get a result every time you use this because you're adding a nitrogen fertilizer. So you don't have a way of knowing it's because of the mycorrhizae or just because you've added a really expensive nitrogen fertilizer. So that's why I say I just wouldn't spend my money on these things, with apologies to anyone who's here that sells these, and I'm not trying to cut into your livelihood, but just from a scientific viewpoint. But here's where you could use them and maybe have an effect that's useful. If you are growing container plants and using a sterilized media, this might be a way to kind of jumpstart the beneficial microbes in that media. So I think there is a use for these products, but not in the landscape. So as I mentioned yesterday, I've got a fact sheet on this, on mycorrhizae. I don't have one on the probiotics, but I have one on the mycorrhizae, understanding how they work and how to nurture them. And I would suggest, whenever possible, think about your systems. Explain to your client that a monocultural agricultural crop is not the same thing as a landscape. The goals are different. The management is different. One is intensive and one is very low maintenance and very sustainable. Use your soil test results to guide fertilizer use. If you've got, as I mentioned yesterday, if you have enough fertilizer, if it's optimum, don't need to add any more. If it's a little bit low, unless you can see evidence of a deficiency in the landscape, do your ground truthing, don't add any of it. If it's excessive, don't add anything. You're saving your clients a lot of heartache and a lot of money, actually, by not having to buy all these products that they really just make their problems worse. Don't amend the soil. And I cannot overemphasize the importance of not adding stuff. Don't add anything as a fertilizer unless you've got the soil test, obviously. Don't add organic material as an amendment. Put it on top. Don't add soil conditioners. They don't work. Don't add packaged microbes. Don't add hydrogels. And don't add biostimulants. There's just no science behind these to show they're going to help in terms of creating a healthy soil environment, especially in terms of today's topic, which is looking at the microbial community. Avoid any unnecessary disturbance. There's that chunky ped. That's what you want. You want those messy-looking clods of soil that we seem determined to grind up. We don't want to grind them up. So we don't want to do this. This has got to be my all-time favorite picture. It was from a landscaping company. And I always say to people, so what happens with those camellia bushes when you're putting the rototiller over that landscape? What happens? Roots are gone, right? Just can't do it. If you have a landscape, you just can't till because you're destroying not just those nice peds, that nice cell structure, but destroying the roots of the very trees and shrubs that you're trying to retain. Now think about, in transplanting, what's in the container and what's in the B&B. Okay, containers have soilless media and B&B has clay. What's in the backfill? Okay, your backfill is native soil. Container media and clay is not the same as your backfill. When you add this as, so when you're putting in an undisturbed root ball from a pot or from B&B, you're putting in a soil texture is completely different than the surrounding soil. This is a barrier. It's a barrier to root growth. Think about where the root crown is. If it's buried in the pot, it's not the plant's fault. And a lot of people are told at the nursery, make sure you're planting it at grade, and they say grade is where it's in the pot, which is not true. If you bare root trees and shrubs, you will find the root crown, and usually it's much lower than it was in the pot or in the B&B. How about the roots? What condition are they in? If they're woody and they're tangled, they're not going to straighten out. If they look like a giant radish, they're not all of a sudden look like a healthy root system. They're just not good roots. Talked about ball and socket and hand and glove yesterday. Again, thinking about how things move and how they stabilize. Hands in a glove are stabilized by the glove. A ball and a socket is meant to move around. And you're going to have to stake things if you plant them without bare rooting them. So think about that. And again, this is not the topic of this talk, although you can tell that I love talking about it. But just to think about when you're trying to have a functional root system during planting as well as after it's established. So what do you add to the planting hole? You add the backfill and the roots and water. That's it, nothing else. What do you add to the planting site? The two miracle products. Some people have already heard me talk about this, and if you talk to Dr. Jim Downer, he will give you the same answer. What are the two miracle products? Water and mulch. And you can talk to any physiologist who works with landscapes, and they will give you the same answer, because those are the two things that trees really, really need. So trees, regardless of where they are, need to have a woody mulch because the mycorrhizae require it, and because it just is a good source of nutrients. And it's what trees have, most trees have evolved under, is something with a woody duff layer. So wood chips are low and slow in nutrient release. They have greater benefits than any other mulches tested. They're just the great choice. With mycorrhizae, the spores are in the wood chip mulches. The wood chip mulches are critical for their growth as active mycorrhizae because the fungal hyphae infiltrate and stabilize the wood chips. Finally, I do need to talk about fire because a lot of people are worried that wood chips are flammable. And wood chips are the least flammable of all the organic mulches because they hold water. And there's been a lot of testing done on this, on the flammability. The one that's burning brightly there, that's rubber mulch. Don't ever use rubber mulch. It's really bad stuff. Arborist wood chip mulch does not change either soil nitrogen or pH. It will change it at the surface where it's touching, but does not change the underlying pH or nitrogen content, so it does not deplete nitrogen resources. And the active fungal communities in there are not pathogens. They're beneficials, maybe as beneficial as being mycorrhizal, certainly nothing that's going to damage the health of the tree. So arborist wood chips are just for so many reasons the right material to use as a mulch if you are growing trees, regardless of the environment you're growing them in. So I have that fact sheet on arborist wood chips. And as I mentioned yesterday, I'm not going to show you all those websites because I talked about them yesterday, but at the bottom of the sheet, there is a link to the ResearchGate library for me. And you can find all of these things as free downloads and more. And with that, I will happily take some questions.
I think we have time for one question.
And then you can also answer me during lunch. I'll be here all day.
So Linda's going to
Yeah.
be at the speaker's table with books and answers.
Yep.
So question.
What about pecan shells for mulch? What do they do? What do they don't do?
Good question. So I know pecan shells are a really popular thing to use as mulch down here. They don't absorb water, so that's the bad part. They can't really be used very much by mycorrhizae or serve as a food source, but they are organic. They do give some structure. I really like using them as like where you have foot traffic and things where you need to have some stability. And so I always suggest to people that they mix their mulches. You know, use wood chips and I think we have hazelnut shells, walnut shells, pecan shells. All those things are great mulches for specific use. And you can make it look really cool and pretty too, depending on how you put them in. You bet. Like I said, I will be happy to answer more questions later. This will work.
Thank you, Linda. And a brief word. I think, to quote from Andrew, we're all partners in chlorophyll, and putting together all of this information, the complex actions and interactions and associations that we see and have learned about and heard about, in order to have health healthy, safer, better trees and landscapes. God, it's really great, and I'm so glad to see everybody here, and I wish I would see some of you joining our Think Trees board, organizational board, to organize next year's conference. And thank you, Linda. We're building a tree one cookie at a time. Thank you, Linda.